And the structure of your paper will be soft question for 16%. So there will be 5 soft questions with 12 marks each, if I'm not wrong. Okay, and then there will be long question, and this is you can choose on it. There will be 3 questions each with 20 marks. Then you can pick 2 of them and then just finish 2 of them. But the short question there will be 5 if I'm not wrong. Then you have to finish everything for the short question part. Okay, so we will now go through the whole thing again. So I have some handouts for you. There is 2 pages for one. So actually this is just a layout for everything. Okay, so that includes basically everything. So you don't need to think that is like the tips here. No, that is just everything. And we will go through it briefly. So I have to turn on the PowerPoint. So first of all, this is like a recap of what we have go through. So we will go through it quickly. So to refresh your memory. So for lecture 1, we'll talk about genes, genome, and genetic. Let me check my, I have to choose this. Okay, so we talk about genes, genomes, and genetics. Then of course you have to have some basic knowledge on what are they, and then some characteristics. What we have covered in quiz, basically they won't be asked again in the exact same way. Okay, so basically if you have answered the question in quiz, I don't think they will happen again in the examination. But if you can still remember what you have answered in the quiz. Okay, so for the gene, of course you have to know about what is gene, and some difference between the genomes or genetic materials in the prokaryotes and eukaryotes. So the basic thing, the complementation in eukaryotes, the membrane, and the membrane mainly decide whether there is a nucleus or not, and whether the chromosome will be concentrated in a region or not. And it also affects where the transcription can happen because of such difference between prokaryotes and eukaryotes. So this major difference, you have to have some idea on it. And then for, still about the structure, so the nucleus, and then the structure of the chromosomes, which are found in, of course, many species. So the chromosome and then the structure, the DNA and RNA, so you have been tested on the difference between DNA and RNA, I guess. But you better still know about their structure, their basic components, and what will each part form the function in the DNA. So the backbone, the nucleotide that make them with ATCG, with diversity. So this is the basic thing about the nucleotides, about the genes, about their structure, function, something like that. Okay, so these are very basic things we have all covered before. Still the structure, and still the structure of the DNA. So I think that you should have remember. And then a bit about RNA. And so this part will happen again and again in your whole course. So you have to know what are transcription, translation, differences, similarity, components involved, how they are different in prokaryotes and eukaryotes, so some major idea like that, where did they happen. And then for RNA processing, it is found only in eukaryotes, and why, and what does it matter, how does it affect the step before and behind. Okay, so this is what we always call the central dogma. This forms the gene expression of DNA to make a protein. Okay, so still about the RNA. So basic feature, chromosome, the wrapping, we have quiz about this, the histones function, the wrapping, mainly to condense the DNA so that they can all be put into a cell. So we have quiz about it already. So this is also covered, coiling, some enzyme to help control the coiling level of the DNA. Yeah, and then about plasmid. So the plasmids, this is some brief introduction of plasmids. So they will happen again and again in later parts because we use plasmid for putting some gene into other organisms or not. So here are the basic thing about plasmid first. You don't have to go really that detail into different types, or just some basic idea of what is a plasmid, their DNA, where are they found, they are small soft segment of DNA, then that will be fine. I won't answer you. Okay, and then the genetic flow. So again, DNA, from DNA to protein. So we always say that it starts from transcription and then translation, maybe with RNA processing in between. But don't forget that we also have DNA replication. Okay, so for the genetic information to be passed from generation to generation, they first have to pass within the cell or within the generation. So there are DNA replication, so don't forget this step. Okay, this is also an important step involved in the whole genetic information flow. So for replication, they duplicate the DNA, making 2 exact copies. So the accuracy rate have to be very high, because you want to make sure every copy is the same, and then you can use the same copy to make protein in later transcription and translation, and also the DNA involved. So basically, these 3 stages you must be familiar with. What are they? Where do they happen? What enzyme is involved? Raw material, products, something like that. Okay, so these 3 major steps in genetic information flow. Yeah, so this introduce the DNA replication here. So just some basic ideas, some key terms. They are semi-conservative, they replicate, make a copy. The raw material, the template is the DNA themselves, and then the product is another exact copy of DNA. Why are they so accurate? Because they have some enzyme to do the proofreading, something like that. Yeah, and the step and the enzyme involved. So basically, enzyme involved in all the important steps. So some key enzyme, if you can tell, like this is using the helicase to unwind it, and then there is a polymerase to add the nucleotide. So some key enzyme involved in each step you have to be familiar with. Okay, so we have quizzed about this leading strand, lagging strand, I think. So it has been covered before. The orchid-summer fragment, why one strand can continuously be made, but the other have to stop and start again and stop, and then link up together. So that is a reason there, but we have quizzed you about that. If you forgot about it, just take a look at the video to show you again. Okay, so as we mentioned, the error rate is very low, or you can say the accuracy is very high in DNA replication. So it is because we have proofreading mechanism provided by the enzyme, also covered in quiz, I think. Yeah, so that is all the point in next one.